What really happened when Earth was the kingdom of giant mushrooms? Dear traveler, greetings. Today we unveil an unrecognizable Earth, covered with immense silent forests, where strange colossi rise. Mushrooms as tall as buildings, dominating plains and shores. A world of shadow and humidity, where vegetation has not yet conquered the sky, and where these fungal giants reign supreme over the landscapes. But before setting off on a new adventure, don't forget to like the video and subscribe to the channel so you don't miss anything. Thank you, and have a nice trip. Between 1.6 and 1 billion years ago, Earth had a very different appearance. The continents were not yet formed as they are today, and life was beginning to organize beyond simple, isolated cells. In this primitive world, the ancestors of fungi appeared, still mysterious. To understand their origins, we must first introduce the supergroup Opisthokonta and its distinction from other eukaryotes. The term Opisthokonta refers to a vast group of eukaryotes sharing a common trait: the presence, at some point in their life cycle, of a cell with a posterior flagellum, sometimes retained, sometimes lost over the course of evolution. It includes animals and fungi. Among them, nucleariids, close to fungi, coexist with other lineages leading to the first ancestors of multicellular animals. To understand what distinguishes a nuclearide from a nascent animal, one must examine their cellular organization and modes of feeding. Nuclearides, unicellular, project fine extensions to capture bacteria and other microprey. The precursor animals already sketch out strategies for multicellularity, though still rudimentary. This divergence traces two evolutionary paths, one towards fungi, the other towards the progressive complexification of the animal kingdom. A key element of fungal differentiation is the appearance of the chitinous cell wall, a resistant polymer offering both protection and flexibility. In the Neoproterozoic ocean, this innovation provides a clear advantage to organisms facing variations in temperature, salinity, and pressure. A true evolutionary milestone, it marks the beginning of the lineage leading to fungi. The first mitochondrial endosymbiosis occurs when an ancestral eukaryotic cell incorporates an aerobic bacterium, which becomes a source of internal energy. The mitochondria thus created transform oxygen into adenosine triphosphate, ATP, the fuel for vital processes. This partnership allows future fungi to better exploit their environment and colonize new niches, promoting their expansion and diversification. At the time, the Neoproterozoic ocean differed radically from today's seas. Vast microbial mats, made of cyanobacteria and other microbes, blanket the seabed and submerged rocks. Oxygen producers, they create microenvironments rich in organic matter. For the earliest fungi or close forms, this abundance of nutrients is an opportunity. They colonize these communities, taking advantage of the chemical interactions that animate them. In this context, the first rudiments of fungal physiology emerge as these organisms, halfway between protists and future fungi, specialize in the partial degradation of organic matter. Bacterial mats, rich in nutrients, serve as their testing ground. While bacteria produce oxygen and nutritional compounds, the protofungi absorb, transform, and store energy in a fragile coexistence where everyone competes for the same resources. In this environment, fungal precursors already possess a solid cell wall and the ability to degrade complex substrates. Some feed on biomass transformed by bacteria, while others develop rudimentary enzymes to break down more resistant molecules. This arsenal allows them to exploit resources inaccessible to other marine microorganisms and to acquire resilience in the face of strong competition. Over time, the distance between nuclearide lineages and animal ancestors widens. The former, close to fungi, retain protist traits, such as the absence of a thick wall and the use of extensions to capture their prey. The latter embark on the journey toward multicellularity, forming more complex aggregates. Thus, some will remain unicellular, while others will begin the great animal adventure. The arrangement of the continents, still fragmentary, influences the evolution of life. Grouped into supercontinents, land masses alter currents and nutrient distribution. In this context, organisms capable of managing resource variations thrive. Protofungi, equipped with mitochondria and early enzymes, colonize both microbial mats and deeper underwater zones. In these aquatic environments, competition is not solely among bacterial mats. Primitive algae and other protists also populate the waters, seeking light or organic matter. Thanks to mitochondrial respiration, protofungi exploit the organic deposits on submerged surfaces more efficiently. They become ecological regulators, recycling debris and supporting a burgeoning network of interactions. Current data indicates that these early fungal life forms remained strictly aquatic for a long time. They often exhibited, at some point in their cycle, a flagellated cell capable of moving towards new microbial mats. Some microscopic fossils, containing traces of chitin, suggest that these aquatic organisms belong to the lineage leading to fungi. Over time, the cellular structure of protofungi strengthens. Genes related to enzyme production multiply, diversifying their metabolic capabilities. Some can thus break down larger molecules, broadening their food sources. Chitin also evolves, combining with other components to solidify the cell wall, a major advantage against marine fluctuations caused by glaciations or warming. Although fragmentary, geological records contain evidence of fungal activity in the form of microfossils or chemical residues. Advanced microscopy and isotopic analysis detect markers related to chitin or certain membrane fatty acids. For some researchers, fungal divergence may be older than estimated. Others remind us of the challenges in interpreting such altered fossils. Thus concludes the initial stage of fungal history, marked by the separation from other Opisthoconta and the acquisition of chitin and mitochondria. Despite the mystery surrounding these precursors, their innovations paved the way for the emergence of more complex organisms. Their aquatic past forges a remarkable adaptability, key to their future conquest of terrestrial environments and the development of the fungal kingdom. Current data shows that the history of fungi is rooted in the protist past, when nuclearides and animal ancestors diverged. Chitinous walls and efficient mitochondria offered them an advantage in changing seas, rich in bacterial mats. Between adaptations and innovations, this stage inaugurates the fungal diversity to come, a prelude to the symbioses and terrestrial mycelium of subsequent eras. Between approximately 720 and 541 million years before our time, Earth experienced a decisive phase called the Cryogenian Ediacaran interval. It marks the end of the Precambrian. Life was then concentrated in the oceans, populated by microorganisms. Our planet was undergoing intense climatic fluctuations, punctuated by glaciations so severe that some scientists speak of a snowball Earth, almost entirely covered in ice even in low latitudes. Despite this hostile environment, microbial mats thrived in the seas. Composed of microscopic algae and bacteria, they covered the marine sediments. Within these biofilms appeared the first aquatic fungi, whose presence gradually transformed the balance among life forms. Microbial mats are aggregates of tiny organisms capable of regrouping and adhering to the shallow seabed. In science, they are termed biofilms. They gather a great diversity of life, including bacteria that break down organic matter and release essential nutrients to other creatures. It is in this environment that aquatic fungi appeared. At the dawn of the Cryogenian, competition for organic carbon, the primary energy source for microbes, became decisive. Long dominated by bacteria, this resource was now contested by fungi also capable of extracting vital compounds. These primitive molds, sometimes with complex names, took an active role in the biofilms, implanting themselves on the surface and feeding on organic debris. Under the ice and within the sediments, this rivalry fueled new survival strategies. In this underwater universe, a particular branch of fungi draws attention. These are the chytrids, Chytridiomycota, their scientific name. Microscopic, they are distinguished by a flagellum, a flexible filament that allows them to move in the water, a rare trait among usually immobile fungi. The chytrids would attach to small planktonic algae, from which they exploited nutrients, sometimes adopting a parasitic mode that gradually weakened their host. This interaction illustrates the intense competition for organic matter. In response, the algae would develop stronger cell walls, or modify their life cycle to protect themselves. This period, marked by powerful glacial cycles, saw the oceans being covered and then freed from ice, disrupting marine ecosystems. When the water approached 0 degrees, aquatic life slowed significantly. But as soon as a thaw occurred, microorganisms took advantage of these brief windows to multiply in microbial mats. Aquatic fungi, thanks to enzymes capable of degrading various organic compounds, then exploited the niches abandoned by other species. Over these freeze-thaw alternations, chytrids and other primitive fungi expanded their distribution, colonizing new shallow marine sites. They interacted with bacteria and algae and reinforced their role in the transformation of organic matter and the circulation of essential nutrients. The study of ancient rocks reveals microfossils called acritarchs. These are small spherical or oval structures, too discrete to be definitively attached to a known biological group. Dating back several hundred million years, they provide a glimpse into the microbial diversity of the time. Some researchers believe that these acritarchs could include spores or forms related to early aquatic fungi like chytrids. Some fossils show thin envelopes reminiscent of chitin. This hypothesis remains debated, as other organisms, notably algae, also produce resistant envelopes. However, their repeated discovery in marine environments suggests that fungi may have occupied a position earlier than previously thought in Precambrian ecosystems. Beyond fossils, one must remember the ecological importance of the first fungal incursions into marine biofilms. Thanks to their varied enzymes, fungi actively contributed to the recycling of organic matter. They could decompose resistant compounds, like certain polysaccharides produced by algae. In return, bacteria and other microbes fragmented the matter into simpler molecules, directly assimilable by fungi. This system of exchanges enhanced the efficiency of biofilms in exploiting resources. Additionally, the degradation of biomass released minerals such as phosphorus, iron, or sulfur, re-injected into geochemical cycles. Thus, the fundamental roles of fungi in the transformation of ecosystems were beginning to take shape. The diversification of aquatic fungi took place in an unstable environment, paced by the freeze and thaw cycles of the Cryogenian. Algae alternated dormancy and resumption, creating organic reserves that the chytrids promptly exploited. Mobile thanks to their flagella, they colonized algae and biofilms. Some established consortiums with bacteria where each used the byproducts of the other. In this game of opposition and cooperation, the outline of future symbioses that would mark the history of life was emerging. One might wonder how microscopic organisms have left their mark in the fossil record. The sturdiness of their cell walls composed of chitin permitted their preservation. Biofilms trapped particles, formed mineral strata which, once compacted, integrated into the rock. Thus, ancient deposits reveal clues of aquatic fungi. Although controversial, these discoveries lead to a reconsideration of the timeline of their diversification. The Cryogenian Ediacaran period appears as a turning point not only climatic, but also crucial for the emergence of marine fungal biodiversity. Sediments from the Cryogenian and Ediacaran eras show a co-evolution between planktonic algae and aquatic fungi. Under the pressure of chytrids, algae reinforced their defenses, and fungi adapted to them. This molecular duel stimulated evolutionary innovation on both sides. Isotopic variations in marine deposits reflect these changes. The composition of algae and the proliferation of parasites altered the amount of buried organic matter and the circulation of oceanic carbon. These interactions, although microscopic, had planetary effects, heralding the great subsequent transformations of the biosphere. The study of these archives relies on two approaches: the observation of thin sections, where every structure is scrutinized, and geochemistry, which reveals the transformation of organic matter and its fungal signatures. The boundary remains blurred. What is assumed to be a fungus might also be an algae or an unknown organism. Nevertheless, the idea is gaining ground that aquatic fungi contributed to the microbial boom. The Cryogenian Ediacaran would have offered them ideal conditions. Abundant nutrients from organic debris and cycles of glaciation favored their gradual development in marine ecosystems. The conquest of marine microbial mats during the Cryogenian and Ediacaran was a foundational moment for fungi. While glaciers covered the oceans, tiny, flagellated organisms colonized algae and biofilms, competing with bacteria. The first fossil traces of chytrids reveal that this struggle preceded the Cambrian explosion. Under the ice caps, fungal life was already playing a decisive role, laying the groundwork for biological complexity. These discrete but powerful cells began to weave the network of interactions that would permanently transform Earth's ecosystems. At the beginning of the Ordovician, about 485 million years ago, the planet changed radically. Vast, shallow seas covered the continents, rich in microorganisms. The climate remained temperate, oscillating between 20 and 30 degrees near the equator. The forming supercontinents created long coastlines where waves deposited sediments and nutrients. Aquatic life diversified there, while the emerging lands, still almost empty, began their slow conquest by new living beings. The dry land then hosted only microbes and cyanobacterial mats. Yet, on the damp shores, some aquatic fungi were initiating a decisive adaptation, leaving the water. Originating from ancient protists with energy-producing mitochondria, they tested new survival strategies. Not yet entirely terrestrial, they thrived in water-saturated substrates at the edge of sediments. This discrete presence already heralded the great transition to terrestrial life, a stage that would upend the history of planetary ecosystems. Evolution offers fungi a major asset, hyphae. These filaments, by intertwining, form the mycelium, a network capable of exploring the soil to capture water and nutrients. Gradually, it ventures into areas alternating humidity and dryness, such as shores and banks. Water regulation mechanisms develop, ensuring survival during dry periods. This setup transforms the mycelium into a foundation for the robustness and diversification of terrestrial fungi, paving the way for their evolutionary success. Parallel to their quest for autonomy, fungi establish a symbiosis with photosynthetic organisms, such as green algae and cyanobacteria. This association, called lichen-like, resembles modern lichens, where a fungus joins with a phototrophic cell. As early as the Ordovician, these precursors colonized bare rocks, benefiting from the production of carbon compounds by their partners. In exchange, the fungal network offers them protection and moisture retention. From this collaboration arose a novel ecology, consolidating the initial steps of land colonization. Fossils from the Ordovician to the Siluran reveal the existence of early mycelial networks. In fluvio-deltaic environments, fossilized filaments appear in the rock, sometimes associated with small pellets identified as microbial coprolites, traces of excrement from microorganisms or primitive arthropods. These clues indicate that a circulation of organic matter already existed, initiating a terrestrial food chain. Thus, fungi played a decisive ecological role long before the establishment of the first complex plants, participating in the dynamics that would durably transform terrestrial ecosystems. The birth of the first soils, or primary pedogenesis, precedes complex plants. At that time, the soil was just an assemblage of broken rocks and forming clays. Fungi, through their hyphae, infiltrated the cracks and secreted organic acids that attacked the stone. This process released essential nutrients, including phosphorus and iron. Phosphorus served to build membranes and transfer energy, while iron supported respiration and metabolic reactions, laying the foundations for more complex ecosystems. The release of phosphorus and iron by fungi disrupts the early lands. Once accessible only in water, these nutrients are now found in the soil, facilitating the establishment of bacteria and rudimentary plants. Fungi thus become architects of ecosystems, altering the soil chemistry and generating new habitats. This dynamic stimulates specialized microbial populations, which enhance recycling and increase the complexity of emerging environments. This silent revolution lays the foundation for future terrestrial ecosystems and prepares the ground for the emergence of the first forests. During the Ordovician-Silurian transition, landscapes were dominated by mineral plains along rivers and deltas, where a few wetlands hosted pioneering algae. Fungi associated with algae or cyanobacteria were the first terrestrial colonizers. Their resistance to drought and recovery upon the return of moisture foreshadowed future lichens. By consolidating the surface with their filaments and then penetrating the substrate, they initiated the formation of a primitive soil, capable of retaining water and trapping particles, thus laying the groundwork for emerging terrestrial ecosystems. Between 485 and 419 million years ago, the terrestrial conquest progressed slowly. Fungi paved the way, but in association with other organisms. They decomposed organic matter while forming symbioses with phototrophic cells that provided them with carbon resources. This strategy ensured their expansion and created favorable conditions for the arrival of more complex organisms. Soon, these fungal networks would see the appearance of the first terrestrial plants, a sign that the transition to more diversified ecosystems was underway. Fossil imprints of mycelia are sometimes visible only under a microscope, in thin sections of sedimentary rocks. They are recognizable by their branched filaments and the chemical traces of ancient fungal matter. Nearby, microbial coprolites attest to small communities where fungi and microorganisms fed on filaments and debris. These clues already reveal the beginnings of terrestrial food networks, at a time when plants were absent. These discrete but fundamental communities laid the first foundations of terrestrial life and paved the way for richer ecosystems. During the Ordovician-Silurian transition, the climate alternated between warming and cooling. A few degrees less were enough to form polar ice caps, while the retreat of the seas freed lands suitable for fungi. In fluvio-deltaic areas, water became intermittent. A brief flood irrigated, then the receding water gave way to drought. Fungi responded by producing resistant spores, capable of surviving until the return of moisture. This adaptation to climatic cycles forged their resilience and confirmed their major role in the first terrestrial ecosystems. The first mycelia, drivers of primary pedogenesis, reveal life's ability to thrive anywhere. Emerging lichens, a union between fungi and algae or cyanobacteria, already illustrate the power of cooperation. The algae produce carbon-based nutrients, while the fungus ensures protection and moisture. This symbiosis, far from being static, diversifies and gives rise to numerous evolutionary pathways that extend well beyond the Silurian. By comparing the chemistry of ancient rocks with that of modern lichens, researchers are gradually deciphering these foundational mechanisms, witnesses to the first steps of organized terrestrial life. In addition to consolidating the soil, mycelia and lichen-like symbioses promote carbon stabilization. The first terrestrial organisms incorporate it into their tissues or bury it in organic form. This storage, though still limited, initiates a dynamic that will intensify with higher plants. Already, fungi are participating in the cycle's regulation. Indeed, their filaments create micropockets of moisture where life develops. Over time, this action transforms the Earth's surface and prepares a more suitable soil for the establishment of the first complex plants. Between the Ordovician and the Silurian, the first mycelial networks and lichen-like associations transform the continents. Enriching the soils with iron and phosphorus, they create an environment conducive to new forms of life. Slowly, the fungi shape the Earth's surface and prepare for the arrival of richer biodiversity. When the first truly structured plants emerge, they benefit from a substrate already modified, facilitating their conquest of the continents. It is these discrete pioneers, by attacking the bare soils, that allowed the Earth to turn green and enter a new phase of its evolution. Prototaxites is an enigmatic organism that lived between 425 and 370 million years ago, from the Silurian to the Devonian. At that time, The emerged lands did not yet host arboreal forests. Vascular plants, barely having appeared, did not exceed a few centimeters. The landscape was thus very different from today. In this minimalist decor, Prototaxites emerged as a conical column reaching up to 8 meters high. For a cryptogam, this size was exceptional. Imagine a ground covered with low vegetation, dominated by these giant silhouettes, heralding a fungal forest before the era of trees. Cryptogam refers to a group of organisms such as ferns, mosses, algae, and fungi, whose reproduction is discrete, often ensured by spores. The word comes from Greek, crypto, meaning hidden, and game, meaning gamete. Unlike flowering plants or conifers, they do not produce visible seeds or pollen. Prototaxites embodies the enigma. With its giant trunk-like appearance, massive structure, and probable fungal or lichenous nature, it rose well above vegetation that barely reached knee height. This recalls the uniqueness of ancient life forms. Prototaxites owes its name to its external appearance. First discovered as a possible fossilized trunk, it was long interpreted as an ancestral conifer, its trunk-like shape reminiscent of a rudimentary pine. However, anatomical studies revealed hyphae. Entwining, they composed a dense mycelium, organized in successive layers, perceptible in cross-section. This structure, incompatible with that of a tree, led researchers to reconsider their hypothesis. Prototaxites was likely not a plant, but an extraordinary fungal organism, a giant fungus of ancient times. Prototaxites fossils have provided isotopic analyses, notably on delta-13 carbon, which compares different carbon ratios. These measurements are used to identify whether an organism fed on marine or terrestrial materials or produced its own food through photosynthesis. The obtained results remain ambiguous, fitting neither a strict plant profile nor a fungal one. Some researchers see the trace of a decomposing fungus absorbing existing material. Others think of a giant lichen, where a fungal part might have housed photosynthetic partners. Two scenarios are opposed. If Prototaxites was a fungus, it would have played the role of a pioneer decomposer, capable of exploiting plant matter and nascent wood. Its giant size would have allowed it to capture more resources and rise for functions, perhaps reproductive, still mysterious. The other hypothesis makes it a giant lichen, a fruit of symbiosis between fungus and photosynthetic organism. Rising in columns, it would have benefited from an almost empty landscape to capture light and grow without competition. Whatever the answer, Prototaxites remains unique. No current lichen approaches such height. Prototaxites appeared as a pyramidal or cylindrical trunk with a smoother, rough surface. In cross-section, its interior revealed concentric layers of microscopic filaments, forming an interwoven hyphae anatomy comparable to superimposed sheets. These lamellae, more or less dense, seem linked to a tiered growth, dependent on the environment. Each ring likely reflects seasonal variations or changes in humidity, temperature, or nutrients. The fossil thus retains a biological memory, tracing the organism's life and the changing conditions of its time. In the Silurian and Middle Devonian, the Earth displayed a warm climate, without great polar ice caps. Terrestrial life was in its infancy. Some arthropods ventured on wet shores, and only rudimentary plants with vascular stems occupied the ground. Forests did not yet exist, but miniature mosses and ferns colonized cliffs and swamps. In this barren landscape, Prototaxites, with its gigantic columns, sometimes formed striking alignments. They thus evoke singular fungal forests, witnesses of a world in full transition towards biological complexity. The expression fungal forests refers to these landscapes where Prototaxites, with its colossal size, dominated the low vegetation made of mosses or small bushes. Imagine a few columns standing above this carpet, creating shade and moisture. Primitive arthropods could take advantage of it, finding refuge or coolness near their base. Some researchers even wonder if a small associated fauna depended directly on Prototaxites, exploiting its tissues or sheltering there. Modest insects or primitive scorpions might have used these giants as havens from the sun or predators. The big question remains unresolved. Was Prototaxites a decomposer or a giant lichen? According to the decomposer hypothesis, it fed on primitive wood and organic debris present on the wet soils. Such a fungal mass could have accelerated decomposition and released essential nutrients into the environment, favoring the growth of the first plants on poor lands. But this reading raises doubts. The analyzed carbon isotopes do not always match known fungal profiles, and the formation of such imposing columns is rare among modern fungi, even if some polypores already produce massive structures. The other vision, that of the giant lichen, relies on the isotopes. Their profiles would resemble photosynthetic activity. A lichen unites a fungus and an alga, or a cyanobacterium. The first provides structure and moisture. The second produces shared organic matter. One could thus imagine Prototaxites as a colossal symbiosis, taking advantage of a world almost devoid of rival vegetation to grow to unprecedented sizes. After all, lichens are capable of invading sterile grounds and resisting extreme climates. In a nutrient-poor Silurian Devonian, such a form could have thrived without relying on wood. The question of internal fossil algae remains, difficult to identify in preserved samples. Even though the mystery remains, most specialists today lean towards a fungal origin, an organism formed from a dense network of hyphae rather than a true lichen. But in science, nothing is ever set in stone. A dominant hypothesis can evolve with new discoveries or more refined analyses. Moreover, the Prototaxites fossils found on several continents do not all deliver the same signature. Some clearly evoke fungi, while others hint at traces of symbiosis. It is even possible that the name Prototaxites actually includes several distinct organisms sharing a similar morphology. Once erected, these giant trunks undoubtedly altered the flow of water. By intercepting rain and dew, they maintained true pockets of moisture at the base of their column. These microclimates favored the development of small fungi and primitive plants, grouped in their shadow. Unlike plants, Prototaxites did not have true roots. Its equivalent was a vast mycelium, an underground network of hyphae that infiltrated the soil to capture nutrients. This dense network also played a role as cement, limiting erosion by water or wind. In this way, Prototaxites was not just a solitary giant. It also contributed to stabilizing the lands and creating underground networks comparable to those still observed in some modern fungi. Prototaxites was probably a pioneer, capable of rising in environments where few organisms survived. As it decomposed, its immense biomass released nutrients that enriched the soils. This influx favored the establishment of other plants, which in turn diversified. Gradually, this process paved the way for the emergence of taller plants equipped with true lignified stems until the first vascular forests dominated the landscapes. But one key question remains. Was Prototaxites a decisive driver of this transition, or merely a secondary player in the evolution of terrestrial ecosystems? The study of fossils also reveals traces of channels or pores in the structure of Prototaxites, which may have transported water and nutrients along its column. According to the fungal hypothesis, these conduits were used for the transport of fluids and enzymes essential for the decomposition of organic matter. According to the lichen hypothesis, they facilitated the movement of water from the outside to the photosynthetic cells. In both cases, such an internal exchange system was probably crucial to support the growth of this extraordinary organism, whose size far exceeded that of other contemporary terrestrial life forms. Approaching Prototaxites also recalls the amazement aroused by the distribution and abundance of its fossils. As paleontologists unearth new segments, they gradually reveal the extent of its range and its adaptability. Geological formations from the late Silurian and early Devonian reveal its remnants in environments near ancient shores or floodplains, suggesting a preference for humid environments rich in organic deposits. Over time, as water bodies and sedimentary cycles shifted, the organism would become buried, offering fossilization conditions the opportunity to preserve it. These mineral layers, true archives of time, retain traces of this giant enigma and allow researchers to piece together bit by bit the forgotten epic of Prototaxites. Faced with these massive trunks woven with hyphae, one question arises: how did Prototaxites ensure its reproduction? In current fungi, fructification takes the form of caps or spongy structures that release spores, while lichens sometimes propagate through small fragments that combine both the fungal part and the photosynthetic cell. For Prototaxites, hypotheses suggest dissemination by wind, rain, or even through the agency of small terrestrial animals, but no fossil has yet provided a trace of a clearly identifiable spore release structure. This silence in the archives remains one of the most troubling aspects of the debate on the true nature of this giant organism. Around Prototaxites evolved an emerging terrestrial fauna, the many segmented myriapods, primitive arachnids, reminiscent of the ancestors of scorpions or spiders, and a few rudimentary insects. No direct evidence shows they exploited the organism, but it is plausible that a trunk several meters high offered much more than a mere solitary silhouette. Its crevices could house a humid microclimate, rich in organic debris, serving as both shelter and a food source. In this still experimental setting of terrestrial life, Prototaxites likely constituted a habitat island, quietly contributing to the early ecological networks of the Devonian. The idea of a wood decomposer, however, raises an essential question. Was there enough lignified material to support an organism of this size? Certainly, in the Silurian and Devonian periods, dense forests did not yet exist, but several plants already had semi-lignified stems or fibrous barks capable of piling up into true organic heaps. These deposits could provide a rich substrate, especially since they were joined by the remains of non-lignified plants, biofilms, and dried algae, invisible today in the fossil records. In the coastal plains and lagoon margins where these materials accumulated, a giant fungus like Prototaxites would not have lacked resources. The image that emerges is less that of a starving giant and more that of a central player in the organic recycling of Devonian landscapes. If Prototaxites was a lichen, its survival depended directly on light and photosynthesis. To reach such a size, it must have benefited from regular sunlight and stable climatic conditions. However, modern lichens grow extremely slowly, which suggests that a giant of this type would have taken decades to rise, patiently accumulating successive layers. Nothing, however, would have prevented it. Large terrestrial herbivores did not yet exist, and the rare Devonian arthropods did not possess jaws capable of cutting into such walls. In this context, Prototaxites could stand as a living pillar, dominating the plain for many years without suffering major aggressions. Whatever hypothesis is retained in the future, Prototaxites remains a striking witness to terrestrial prehistory. While plant and animal life was just beginning to venture out of the waters, some organisms were already reaching unexpected dimensions. These gigantic columns dominated a bare, almost desert-like landscape, far from the lushness we know today. One can imagine the few terrestrial creatures of the time bypassing them, seeking a bit of shade or protection from predators. Thus, Prototaxites appears as a true natural monument, standing in the penumbra of a world still in gestation. The influence of Prototaxites on the Silurian and Devonian landscapes was far from negligible. By its size, it disrupted the organization of a still minuscule flora. Disposed at intervals, these giants formed an irregular cover that created at times shaded and protected areas, and at other times more exposed spaces. This mesh, stable over several millennia, would have contributed to transforming the soils and offering refuges to numerous microorganisms. Thus, Prototaxites played a pioneering role. It opened the way for vascular trees and laid the first foundations upon which the diverse great forests of the planet would eventually thrive. Despite decades of analyses, the mystery remains. Researchers rely on more refined techniques in microscopy and organic chemistry, as well as the study of fossil pigments, to detect potential photosynthetic components. Others examine the macromolecules trapped in the substrate to identify enzymes linked to wood decomposition. Each technological advance provides a lead, but the fragility of the remains prevents a complete resolution. Prototaxites thus remains a fascinating enigma, reminding us that in the early days of terrestrial ecosystems, nature was already experimenting with unexpected forms of adaptation. This enigma reminds us that life's expansion did not follow a single path, but multiple ones. Pending a consensus, Prototaxites remains the symbol of the vital forces that were already beginning to shape the continents. In the Middle Devonian, about 400 million years ago, terrestrial life reached a decisive milestone. In this warm and humid world, the first plants established themselves on the emerged soils and forged an unprecedented partnership with microscopic fungi capable of penetrating their tissues. This pact, which we now call mycorrhiza, would durably transform the ecological balance of the continents. The oceans still covered vast expanses, while the moving continents formed lagoons and marshes rich in nutrients. Gradually, shores emerged, offering favorable grounds for primitive plants. It is in this context of geological upheavals that the first durable links between underground fungi and terrestrial plants were formed. Mycorrhizae are associations between primitive roots and fungi capable of exploring the inside or the surface of plant tissues. This mutualism ensures the survival of both partners. The fungus provides the plant with nutrients that are difficult to obtain, particularly phosphate, while the plant returns sugars from photosynthesis. Each offers what it controls and receives what it lacks. This biological exchange has greatly favored plant diversification, especially in poor and unevenly resourced environments. Devonian soils, still thin and poorly mineralized, made this collaboration vital. The fungi, capable of exploring vast underground surfaces, represented a true revolution for early plants. The Rhynie Chert site in northern Scotland offers exceptional fossil evidence of this foundational link. These silicified rocks, formed by the action of volcanic deposits and silica-rich waters, have preserved primitive plants like Aglaophyton. Devoid of true roots and leaves, this species was limited to creeping stems absorbing water and nutrients. However, in these fossilized tissues, researchers identified hyphae, forming small microscopic arbuscules. This discovery attests to the presence of mycorrhizae, and more precisely, endomycorrhizae, where the fungus lives directly inside plant cells. The fossil fungi from Rhynie Chert belong to a group once called Glomeles, now known as Glomeromycetes. Their specificity is the formation of arbuscules, small branches that facilitate the exchange of nutrients. Their extremely fine hyphae extend into the soil to capture phosphate in forms inaccessible to plants, then restitute it in an assimilable form inside plant cells. Before the appearance of mycorrhizae, access to phosphate remained difficult. Indeed, rock minerals were only slowly released through erosion, hindering plant growth. At the time of Aglaophyton, soils remained nutrient poor. Fungi, however, had already developed strategies to explore the substrate and decompose organic matter. Their hyphae penetrated crevices, dissolved certain minerals, and extracted nutrients. Once transmitted to plants, these stimulated photosynthesis and biomass production. In return, plants provided more organic matter, reinforcing fungal nutrition. This virtuous circle quickly favored more complex terrestrial ecosystems. The role of mycorrhizae goes beyond nutrition. By promoting the establishment of more robust plants, they stabilize early soils. Denser vegetation retains humus, enriched by organic debris, and gradually forms a fertile layer capable of storing water and nutrients. Microorganisms thrive there, and a cycle begins. Carbon dioxide is absorbed by the plants, transformed into biomass, and then integrated into the soil as organic carbon. This sequestration gradually modifies the atmospheric balance. In the mid-Devonian, although not yet true forests, the expansion of plants marks a major turning point in carbon cycles and prepares the future transformation of the biosphere. This fungus-plant pact continues to fascinate scientists as modern mycorrhizae extend a model inherited from the Devonian. more than 80% of today's plants still live in association with soil fungi. Only a few specialized families, having adopted other strategies, do without them. This continuity reflects an immense evolutionary success. The nutritional and structural benefits offered by mycorrhizae have been so critical that their abandonment remains exceptional. The lineages that have detached from them had to invent equally elaborate alternative mechanisms. The mid-Devonian, with Aglaophyton, and the glomeromycetes, thus provides one of the oldest fossil evidence of this crucial alliance. The Rhynie Chert site constitutes an exceptional discovery. The silica deposits from hot groundwater there have preserved the organisms with rare precision. These fossils show not only the external appearance of the plants, but also their internal structures, offering researchers a detailed view of the cells. One can clearly distinguish the arbuscules, small branching structures characteristic of endomycorrhizas, which multiply the contacts between fungal hyphae and plant cytoplasm to accelerate nutrient exchange. Thanks to this unique conservation, paleobotanists have been able to reconstruct the functioning of an ancient ecosystem, which, despite its apparent simplicity, was already preparing future biological innovations. The phosphate-sugar exchange involves a whole chain of transformations in the soil. Fungi secrete enzymes that break down organic matter, thereby releasing nitrogen and phosphate, which are immediately transmitted to the plants. These, thanks to photosynthesis, provide sugars in return, which are directly integrated into the fungal filaments, which the fungus metabolizes or stores. This mechanism creates a vast network of subterranean exchanges, comparable to an invisible web connecting plants and fungi. Through this network, each partner benefits from the other's resources, and the entire ecosystem benefits from more efficient feeding and better stability. The advance in the plant-fungus alliance has profoundly marked plant evolution. Thanks to more stable nutrition, plants were able to grow, develop solid supportive tissues, and extend their photosynthetic surfaces. Mycorrhizas undoubtedly played a decisive role in the explosion of plant diversity. The descendants of Aglaophyton, the first vascular plants, and many other emerging lineages benefited from this underground support. Gradually, the emergence of true roots, branching stems, and more efficient leaves increased their adaptability. In parallel, fungi also diversified their own lineages, adjusting their colonization strategies to these increasingly complex partners. The discovery of fossilized mycorrhizas in the Rhynie Chert was pivotal in understanding how terrestrial life crossed an essential threshold. Researchers first identified remarkably preserved fossil plants, but while examining their cells, they highlighted branched fungal hyphae, indisputable traces of mycorrhizal colonization. Seeing this partnership in stone, previously known only in modern plants, was a revelation. This fossil evidence offers science a unique window into nature's ingenuity, capable from its beginnings of optimizing exchanges between organisms to lay the foundations for future terrestrial ecosystems. Researchers have shown that Aglaophyton did not possess true roots, but rather rhizomes, types of underground stems providing anchorage and water transport. The fungus, by colonizing this network, played a role similar to what roots would later assume. Thanks to their hyphae, nutrients were better absorbed, allowing the plant to expand widely. One then imagines the wet Devonian landscapes, traversed by muddy channels and covered with carpets of low vegetation, from which a few creeping or erect structures emerged. Under this greenery, a discrete but essential fungal network already supported the ecosystem's balance. This foundational pact, initially limited to a few species, gradually became the rule for most terrestrial plants. While the phosphate-sugar exchange remains its core, mycorrhizal networks have undoubtedly facilitated the circulation of other molecules, such as hormones or chemical signals. This explains certain coordinated reactions to stress or environmental changes. This symbiosis is flexible. Indeed, when the plant lacks nutrients, the fungus adjusts its support. When it experiences a carbon deficit, the plant increases the sugar supply. More than a simple trade, it is a constant dialogue between two lineages that have co-evolved over hundreds of millions of years. On a global scale, The increased plant biomass through mycorrhizae has contributed to the long-term storage of carbon. With each cycle, plants fixed more carbon dioxide, some of which accumulated in the soil as organic matter, later recycled by various decomposers, including fungi. This process, repeated over long periods, eventually influenced the atmosphere's composition and climate evolution. Certainly, the Middle Devonian did not yet witness the rise of large forests, but the first endomycorrhizae already laid their foundations. The plant world, enriched with nutrients, could then colonize new spaces and give rise to more complex life forms. Without mycorrhizae, plant history would likely have followed a much slower, possibly unfinished trajectory. The fossils of the Rhynie chert testify to this and remind us how cooperation has shaped the biosphere's balances. If the Earth has become so green and rich in plants, it's thanks to these early partnerships established well before the appearance of large forests. Aglaophyton, modest in appearance, lit the spark that would lead to the diversification of the plant kingdom. As for fungi, they early on demonstrated their ability to explore diverse niches and unite with plants to build prosperous communities. In the lower Carboniferous, between 360 and 320 million years ago, Earth hosted vast, warm, and humid forests. In these swamps dominated tree-like lycophytes, similar to today's lycopods, but capable of reaching over 30 meters. The equatorial climate often exceeded 20 degrees Celsius, with abundant rainfall. Massive trunks emerged from a muddy, waterlogged soil, creating a landscape where colossal vegetation rose above flooded expanses as far as the eye could see. Emerging mountain chains, resulting from slow tectonic movements, shaped sedimentary basins where water accumulated with thick plant deposits. Riverbanks and alluvial plains were covered with giant ferns and horsetails, while lepidodendron and sigillaria, imposing lycophytes, dominated the canopy. Their trunks, marked by scale-like patterns, bore the traces of vast leaves. A thick bark protected a tissue rich in lignin, ensuring rigidity and solidity. Lignin refers to a complex assembly of organic molecules. It gives woody plants their rigidity and protects them from microorganisms. Today, some fungi, such as Basidiomycota, including boletes, amanitas, and numerous decomposers, produce enzymes capable of breaking it down, including lignin peroxidases. But in the lower Carboniferous, these fungi did not yet possess these chemical tools, making lignin nearly indestructible. This detail explains the massive accumulation of plant matter that led to the future coal deposits. In the Carboniferous swamps, dead vegetation fell into stagnant waters low in oxygen, slowing decomposition. Debris accumulated in layers, forming peat. Today, various organisms, including wood-decaying fungi, can degrade lignin, but at the time, only cellulose and hemicellulose were attacked. Unable to break down the woody framework, nature saw thick layers of semi-decomposed plant matter stack up, with no effective mechanism to eliminate them completely. Over millennia, the accumulation of plant layers, covered by sediments, compressed the peat and expelled its water. Under pressure and increasing heat, it slowly transformed into coal. This is carbonification. The absence of lignin decomposers allowed for the massive accumulation of plant matter, leading to the vast coal deposits that humanity would exploit intensively during the industrial era. The Carboniferous swamps formed vast, humid forests, dominated by immense lycophytes, tree ferns, and giant horsetails of the genus calamites. Their growth, stimulated by air rich in CO2 and a warm climate, created woods so dense that they retained moisture like a natural greenhouse. Through photosynthesis, these plants stored carbon in their tissues, but decomposition, limited by the wood's composition and the absence of suitable enzymes, slowed its return to the atmosphere. Contrary to the idea of a total absence, many fungi lived alongside Carboniferous plants, degrading cellulose or simple substrates, or developing as molds and aquatic fungi. But Basidiomycota, with peroxidases capable of breaking down lignin, had not yet emerged or spread on land. This gap between the abundance of lignified wood and the absence of efficient recyclers was crucial in the formation of immense coal deposits. Contrary to the idea of a total absence, many fungi lived alongside Carboniferous plants, degrading cellulose or simple substrates, or developing as molds and aquatic fungi. But Basidiomycota, with peroxidases capable of breaking down lignin, had not yet emerged or spread on land. This gap between the abundance of lignified wood and the absence of efficient recyclers was crucial in the formation of immense coal deposits. The woody matter of the Carboniferous swamps contained cellulose, hemicellulose, tannins, and especially lignin, responsible for the rigidity and impermeability of wood. Easier to degrade, cellulose often disappeared before lignin, which remained intact due to the lack of specialized enzymes. Compressed and concentrated, this biomass transformed into solid carbon, peat, then lignite, bituminous coal, and, after millions of years and tectonic pressures, anthracite. In the early Carboniferous, the process was still in its early stages. The fauna of the Carboniferous, quite different from that of the Jurassic, included large terrestrial arthropods, giant millipedes, sizable insects. They thrived due to a high oxygen level, as well as precursor amphibians akin to giant salamanders. In these swampy forests, none of these animals could degrade the lignin of dead wood. They limited themselves to the bark or cellulose, leaving intact the most resistant part of the plant biomass. The Carboniferous is nicknamed the Coal Age due to the plant accumulation linked to the absence of ligninolytic fungi. One might rather speak of a wood age, marked by the massive production of poorly decomposed wood. On Laurussia, which then included parts of North America and Europe, vast swamps left thick layers of bituminous coal, visible today in Great Britain, Germany, France, or the United States. Gondwana, to the south, also experienced local deposits according to the climate. The absence in the Carboniferous of fungi capable of effectively degrading lignin played a decisive role, almost by default. If ligninolytic Basidiomycetes had existed then, most of the wood would have rotted, and carbon would not have accumulated underground. It was only later in evolution that these fungi developed peroxidases and other enzymes capable of breaking down the complex chemical structures of wood. In these swampy forests, trunks lay in the muddy water, quickly covered by new layers of plant debris. The linear leaves of lycophytes formed a thick carpet, while the stagnant, dark water, rich in fulvates and humates, nourished various microorganisms. These degraded the simple compounds, but the lignin persisted, gradually being buried. New shoots settled on these partially buried trunks, creating over millennia a stacking of almost intact vegetative layers. This process changed soil chemistry, the availability of nutrients, and the chemical balance of the swamps. In stagnant waters, some bacteria produced methane from incompletely degraded organic matter. Climatic variations, marine or tectonic movements, could bury these layers deeper, isolating them from oxygen and enhancing sedimentation. Buried under new strata, these deposits underwent increased pressure and heat, initiating their gradual transformation into coal. Scientists reconstruct the history of the Carboniferous thanks to clues preserved in coal mines and fossils. Microscopic examination of thin coal sections reveals spores, barks, and lycophyte tissues, sometimes marked by the diamond patterns unique to Lepidodendron. These observations confirm the central role of lignin. Meanwhile, mycologists note the near absence of fungal structures capable of ensuring advanced wood decomposition, highlighting the role of this lack in plant accumulation. The lower Carboniferous is sometimes described as a time when the planet was investing in the sustainable storage of carbon, waiting for the emergence of more efficient decomposers. This image effectively illustrates the combination of biological and geological processes that produced thick layers of coal, sometimes several meters high, repeated in the strata according to the transgression-regression cycles and the succession of swamp forests. In the Carboniferous swamps, dead wood, often half-submerged, dominated the landscape, while few fungi were capable of breaking it down. Lycophytes formed dense stands, accompanied by some tree ferns. Insects grazed on soft leaves and stems, and large amphibians hunted at the water's edge. The massive production of lignin far exceeded recycling capacities, sealing the fate of these forests, destined for burial and fossilization. This is not just the story of poorly decomposed wood, but an event with major consequences for both terrestrial and human evolution. Millions of years later, the exploitation of coal, born from the absence of ligninolytic peroxidases in the Basidiomycota of the Carboniferous, would fuel the Industrial Revolution. Thus, an ancient enzymatic deficiency shaped, underground, a part of humanity's energy destiny, illustrating how a biological detail can weigh on world history. Today, we know that coal results from natural carbon sequestration during the Carboniferous. By burning it, we release into the atmosphere carbon dioxide that was once buried due to the lack of enzymes capable of breaking down lignin. This geological legacy partially conditions our current emissions. Without this decomposition defect, the Earth, its coal reserves, and the course of human industrial history would have been profoundly different. About 300 million years ago, vast forests and swamps covered a large part of the planet, accumulating enormous amounts of dead biomass and storing carbon in geological layers. These partially decomposed deposits are the precursors of coal. But a major event occurs: the appearance of white rot. This enzymatic innovation, carried by certain basidiomycete fungi, would profoundly transform terrestrial ecosystems and disrupt the balance of wood decomposition. Basidiomycetes form a vast group of fungi, including today's chanterelles, boletes, or agarics, and reproduce through basidia-producing spores. At the time, this group was still diversifying. A particular lineage developed enzymes capable of breaking down wood lignin. This capability, known as white rot, would transform plant decomposition and disrupt the balance of terrestrial ecosystems. Lignin is a robust plant molecule that provides rigidity and support to stems and trunks, allowing plants to grow toward the light. Much more resistant than cellulose, it challenged microorganisms and fungi lacking specialized enzymes, which could only attack simpler components. Thus, enormous amounts of lignin-rich material accumulated in soils and marshes, slowly turning into peat and then into coal. Everything changes when basidiomycetes produce two key enzymes, lignin peroxidase and manganese peroxidase. Capable of breaking the stable chemical bonds of lignin, composed of aromatic ring molecules, they act through oxidation reactions in the presence of oxygen and cofactors. A fungus mastering these tools can finally decompose lignin and access the nutrients in wood, significantly surpassing all other organisms in the competition for this resource. This innovation triggers a true ecological revolution. Basidiomycetes cause white rot, identifiable by the pale appearance of wood, where lignin is consumed first, while cellulose partially remains. Now, lignin becomes an exploitable resource, accelerating and deepening woody decomposition. Nutrients are recycled more efficiently, and soil chemistry evolves, transforming the dynamics of ancient forest ecosystems. The appearance of white rot profoundly changes the long-term storage of carbon. Before, a large portion of woody biomass accumulated intact in sediments, forming thick carbonaceous deposits. Once basidiomycetes manage to degrade lignin, this easily accessible carbon disappears. Dead wood is fragmented, transformed, and recycled more quickly, slowing the formation of new deposits. On a scale of millions of years, geological records reveal a clear decline in the deposit of fossilized carbon and a reorganization of forest ecosystems. Imagine vast forests of tree ferns and primitive seed plants, where enormous woody debris accumulates. The innovative basidiomycetes trigger white rot, relegating microorganisms lacking specific enzymes to a secondary role. This process transforms soil structure, rapidly releases nitrogen, phosphorus, and potassium, and accelerates the renewal of flora. In an imaginary slow motion, a fallen trunk becomes covered with hyphae exuding enzymes, gradually lightening the wood until it is entirely recycled into the cycle of life. At the same time, a co-evolution develops between saprotrophic fungi and xylophagous insects that feed on wood. Among these are primitive cockroaches, ancestors of current forms, and the first beetles. Less diversified than they would be later, some nibble on decomposing plant tissues, others burrow galleries, thus coming into direct contact with the Basidiomycetes. White rot opens new opportunities for xylophagous insects. By pre-degrading lignin and cellulose, it makes the wood softer and easier to chew, for mandibles still not well adapted to hard fibers. By tunneling their galleries into a trunk invaded by mycelium, these insects transport and disperse fungal spores, allowing the fungi to colonize other areas. The two partners thus evolve together in a co-evolution relationship. This co-evolution leads to increasing specialization. Early cockroaches adapt their diet and mouth parts to already degraded wood, while Basidiomycetes refine their chemical strategies. Basal beetles open up to varied woody substrates, from decomposing wood to temporarily submerged trunks. The fungus weakens the wood, the insect feeds on it, and upon leaving, transports spores and mycelial fragments, promoting the ecological expansion of both partners. Imagine a vegetative cover dominated by immense lycophytes and tree ferns. On the ground, fallen logs lie, sometimes submerged in stagnant waters. Heat and humidity encourage the proliferation of fungi, exploiting these waterlogged substrates. Where white rot acts, the soil, quickly recycled, is enriched with humus, stimulating mosses and young ferns. Elsewhere, without these Basidiomycetes, dead wood accumulates, offering a striking contrast of forest dynamics. Furthermore, this ability to decompose lignin changes the chemistry of the atmosphere and oceans. Carbon, once buried for millions of years in the form of peat or coal, is now released more quickly as carbon dioxide through microbial respiration linked to decomposition. Over tens of thousands to millions of years, this surplus influences climate, plant growth, and nutrient circulation, pushing life to adapt or undergo these changes. The geological consequences also appear in sedimentary basins where coal was once developed. With white rot, incompletely mineralized woody material becomes scarce. Coal formations persist, but their accumulation rate slows, and their composition evolves. Fossil records reveal, through the chemistry and texture of deposits, the imprint of this lignin degradation, confirmed by mineralogical and organic indices, attesting to the action of unprecedented fungal enzymes. Certainly, here is the translation. In addition, the chemical functioning of these enzymes deserves to be clarified. Peroxidases, whether lignin peroxidase or manganese peroxidase, use hydrogen peroxide to initiate oxidation reactions capable of breaking the extremely resistant bonds of lignin. Manganese peroxidase acts notably in the presence of manganese ions, which charge and discharge during the reactions, extracting electrons from the targeted molecules. This highly effective process triggers a cascade of chemical breaks that weakens the wood, making it brittle and spongy. Even today, many forest saprotrophic fungi exploit this process, reducing a massive trunk into a soft white mass within a few years. The arrival of Basidiomycete fungi producing white rot marks a turning point in the history of the biosphere. The enzymatic innovation behind white rot illustrates the creativity of the fungal world. For a long time, lignin was a chemical barrier that only a few slow pathways could break. This evolution also diversifies insects. Fossil cockroaches, primitive lignocoles, and basal beetles initiate an expansion that will lead to today's beetles, ladybugs, and ground beetles. Thanks to specialized peroxidases, Basidiomycetes have conquered a previously inaccessible niche, becoming masters of wood decomposition. Appearing between the late Carboniferous and early Permian, this capability emerged in stages with marked evolutionary leaps. Gradually, forest humus is enriched with partially degraded organic residues, also transforming the soil microfauna. This moment connects two issues: carbon storage and biological interactions for recycling material, More than a stage of degradation, white rot modifies the biosphere, influences carbon reservoirs, and diversifies the feeding strategies of insects, from spore transporters to symbionts. In all forests, fungal filaments degrade cellulose and lignin thanks to efficient peroxidases, perpetuating a key role in organic recycling for hundreds of millions of years. By reducing coal formation, white rot has transferred carbon from ancient geological reservoirs to the biosphere and atmosphere, influencing their exchanges sustainably. At the end of the Permian, 252 million years ago, a massive extinction wiped out most species. Forests collapsed, leaving trunks and leaves on depleted soils. Geological records then show a fungal spike, known as the spike fungique in French. This refers to an abundance of fossil spores called Reduvia sporonites from saprotrophic fungi decomposing dead matter. Understanding this phenomenon requires diving into the dramatic context of this planetary crisis. Geological traces show destroyed forests covered with trunks and plant debris. This disaster, caused by volcanic gases, a disrupted climate, and unstable ecosystems, leaves a gigantic reservoir of organic matter. Decomposer fungi exploit this feast, proliferating massively and occupying niches abandoned by plants and other consumers. Reduvia sporonites refers to fossil spores of fungi. Unlike plants, they do not produce seeds, but tiny structures dispersed by wind or water, which germinate into mycelium in soils or debris. Normally rare in the fossil record, they multiply massively at the end of the Permian, marking an extraordinary proliferation. The Earth is covered with a carpet of organic debris. Forests disappear, erosion accelerates, and food chains collapse with the loss of many animal species. Fungi exploit this dead biomass. Large trees deprived of sap become a perfect substrate where their filaments proliferate, releasing countless spores to ensure their survival. At the end of the Permian, more than 90% of marine species and many terrestrial species disappear. Fungi, having become key players, recycle organic matter and enrich the soil with nutrients. But their rapid decomposition also releases large quantities of carbon dioxide, exacerbating the already severe climatic upheavals. Increased decomposition of vegetation promotes the influx of nitrogen and phosphorus into the oceans, potentially triggering eutrophication. At the end of the Permian, these inputs, combined with climatic and volcanic crises, disrupt planktonic productivity. Algal blooms and hypoxia follow, exacerbating marine losses. A vicious circle sets in: extinctions, fungal explosion, release of CO2 and nutrients, alteration of climate and oceans. All this intensifies the crisis. At the end of the Permian, the planet warms by several degrees Celsius. The causes include the massive eruptions of the Siberian traps and the rapid decomposition of terrestrial biomass. The disappearance of forests releases stored carbon, which fungi and bacteria transform into CO2, amplifying the greenhouse effect and trapping more heat in the atmosphere. Behind the magnitude of the extinction lies a reorganization of life. The fungi of the fungal peak, adapted to extreme environments, take advantage of an influx of resources to grow rapidly thanks to enzymes capable of degrading lignin and cellulose. The annihilated forests become an ideal resource, evidenced by the fossil abundance of the spores Reduvia sporonites. The true nature of Reduvia sporonites is debated in the scientific literature. While these spores are generally interpreted as those of saprotrophic fungi, some researchers suggest they may originate from particular algae or other microorganisms. The fungal hypothesis remains largely dominant, supported by the internal morphology of these spores, similar to that of modern decomposer fungi, and by the context of massive deforestation, offering an abundant organic substrate. After a fungal peak, landscapes remain barren, fragile, eroded soils, and layers of decomposing trunks. Fungi colonize wood and soils, weaving vast networks and covering surfaces with thin colored films. Fauna and flora survive with difficulty. Only ferns and resistant plants persist. Dominating the dead matter, fungi recycle carbon, which returns to the atmosphere or dissolves in water, influencing geochemical cycles. The fungal explosion at the end of the Permian leads to a CO2 peak, a gas that, in excess, causes climatic upheavals. Combined with the giant volcanic eruptions, this massive decomposition enhances global warming and plunges Earth into intense environmental stress. The overheated and eroded soils become unstable, delaying plant recolonization. The influx of nutrients into the oceans triggers a bloom. Thus, microalgae and cyanobacteria proliferate massively, but their death leads to a decomposition that depletes oxygen, forming dead zones where marine life disappears. This cascade accentuates animal losses, already weakened by climate and ecosystem destabilization. The fungal peak, a major marker of the Permian crisis, reveals the massive destruction of forests and the rapid colonization by fungi. The rocks of this period contain layers rich in fossil spores, associated with a rise in CO2 and a modification of the carbon isotopic ratio. The analysis of these isotopes allows tracking the sources and cycle of carbon in ancient environments. After the extinction, a part of terrestrial life comes close to definitive disappearance. Balance returns only after millions of years. Dominating the scene, fungi accelerate decomposition and alter the landscape. Their hyphae modify minerals, releasing nutrients, while the absence of roots exposes soils to erosion. This reconfiguration, nevertheless, prepares the ground for the gradual return of new plants and animals. The saprotrophic explosion slows the return of forests. While fungal decomposition provides useful nutrients, it also releases carbon dioxide, worsening the greenhouse effect and climatic instability. Seedlings and young shoots struggle to establish themselves. This fragile balance changes over millions of years, until the progressive resurgence of life, transformed by these upheavals. The Reduvia sporonites testify to a brief period where fungi dominate terrestrial ecosystems. Taking advantage of the disappearance of species and the accumulation of organic matter, they assume the role of cleaners, transforming dead wood and debris into minerals that will nourish future ecosystems. Even after a near-total collapse, life reorganizes. The post-Permian fungal proliferation is proof of this. Indeed, in the chaos, certain organisms prepare the rebirth of ecosystems. The fungal spike fades, carbon cycles stabilize, and in the Triassic, new forests establish themselves, marked by the rise of modern gymnosperms. The study of fossil spores shows how organisms react to major ecological crises. Chemical analyses, morphological observations, and microscopic examinations confirm the Permian fungal peak and trace the events that caused it. Subsequent crises sometimes show comparable fungal signals, but less pronounced. The unique scale of the Permian episode makes it a textbook case, illustrating the chain reactions in a disrupted world. The rapid rise of mycelia and the profusion of fossil spores show how an opportunistic group can transform the planet when ecological locks disappear. Between 252 and 201 million years ago, in the Triassic, the Earth recovers from the Permian extinction. Surviving flora and fauna reorganize within reconstructing ecosystems. As the environment changes, fungi and fungus-like organisms occupy new niches, forcing remaining communities to adapt to a transformed world. The fungal spike of the late Permian diminishes in the Triassic. Debris stabilizes, new plants establish themselves, and fungi adjust their interactions with the surviving flora, ushering in a new era of adaptations. At the beginning of the Triassic, conifers, cycads, and ginkgos colonized the lands thanks to their drought tolerance. Their roots form symbioses with fungi to extract nutrients from poor soil. These new forests provide favorable habitats for the expansion of adapted plants and the rise of innovative fungal strategies. The mixed forests of the Triassic associate gymnosperms with robust ferns, offering multiple niches for fungi. Decomposers of wood, colonizers of roots, or aerial tissues, they participate in a balance that favors coexistence. The plants provide organic matter and shelter, and the fungi, in return, support their nutrition and regulate carbon and mineral cycles. In the Triassic, endophytic fungi, living in plant tissues without damaging them, enhance resistance to heat and drought, optimize nutrient absorption, and neutralize certain toxins. These symbioses make plants more efficient in an unstable climate and would have been crucial for the reconstitution of forests after the Permian crisis. Drought and soil salinity test the plants. Endophytic fungi reduce these constraints by regulating mineral absorption, limiting saline toxicity, and maintaining water balance. These alliances allow certain species to colonize arid environments, enhancing the resilience and expansion of young forest communities. In the Triassic, fungus-like organisms such as oomycetes, close to fungi, gain importance. As phytopathogenic parasites, they attack seeds, roots, or leaves, altering plant composition. Fossils reveal characteristic lesions and deformations. By selecting the most resistant species, these pathogens help shape the structure of forests. Oomycetes, with cell walls rich in cellulose, resemble algae or plants, but remain distinct. In a competitive environment, they employ various strategies: dispersal in moist environments through their water-mobile spores, or using insects to infiltrate damaged tissues. These methods allow them to take advantage of post-Permian ecological disorder. By weakening certain plants, parasites can disrupt the entire plant community. These losses favor the establishment of more resistant species, often associated with symbiotic fungi. The Triassic landscape alternates between devastated zones and regeneration areas, where decomposition enriches the soil with nutrients. The evolution of forests results from this complex interplay of interactions. In the Triassic, plant-fungus cooperation varies depending on the environment. Wet areas favor plant growth and offer numerous substrates for decomposers. In dry regions, only plants associated with resistant endophytes thrive. Parasites and symbioses adjust to evolutionary pressures, forming an unstable balance that strengthens ecosystem resilience. Under the heat of the Triassic, with temperatures exceeding 30 degrees Celsius in some areas, drought persists within the interior of Pangaea. Plants allied with symbiotic fungi dominate, while parasites exploit every weakness. Ecological successions gradually stabilize communities, where adaptive diversity ensures survival. Meanwhile, root interactions intensify. Mycorrhizal fungi help plants capture phosphorus, iron, and other rare elements in poor soil. Fossilized roots preserve these traces, evidence of their key role in ecosystem expansion. The combination of endophytes and mycorrhizae strengthens plant resistance, allowing certain Triassic forests to colonize hostile areas. In these forests, fauna disperse the spores of fungi and oomycetes. Primitive reptiles transport them on their scales or through their droppings, while beetles release nutrients and spores by burrowing into dead wood. These breaches benefit pathogens, but mutualistic plants and fungi strengthen their defenses, establishing a balance between expansion and regeneration. In the Triassic, climate, propagule dispersion, and biological interactions shape ecosystems. The advantage of one species can be the loss of another, but these changes forge resilient biodiversity. Fungi and oomycetes, drivers of diversification, transform landscapes through decomposition, parasitism, or collaboration, preparing the next chapter of Earth's history. Some oomycetes target specific plants, others have many hosts. Their diseases cause die-offs that pave the way for unpredictable vegetative successions. Fossils reveal their impact, while endophytes and mycorrhizae stimulate resistant species. This game of attack and counterattack shapes a cycle where the decline of one species feeds the prosperity of another, redrawing the landscapes. From 201 to 66 million years ago, the Jurassic and Cretaceous see the rise of large conifers and the dominance of dinosaurs. Fungi evolve, with the appearance of entomopathogenic forms and the emergence of boletes and amanitas in symbiosis with trees. In this warm, humid world, they interact with fauna and flora, contributing to ecological balance. In the Jurassic and Cretaceous, a warmer climate than today, punctuated by cooler or drier episodes, shapes species distribution. Vast forests of primitive conifers dominate, reaching remarkable heights. In these forests, immense herbivores consume leaves, branches, stems, ferns, and primitive fruits. Carnivores track their prey, while small omnivores seize every opportunity. Insects in full diversification, beetles, primitive butterflies, ancestors of grasshoppers, giant dragonflies, occupy various niches. Fungi and insects already develop close interactions. In the plains, tree ferns and horsetails surround swamps, creating rich, humid corridors conducive to biodiversity. Probably appearing at the beginning of the Mesozoic, entomopathogenic fungi, akin to cordyceps, infect certain insects and alter their behavior. The host is forced to place itself on a plant or site conducive to spore dispersion. The fungus consumes its resources, alters its brain chemistry, then the insect dies, transformed into a receptacle for spores. Spectacular but essential, entomopathogenic fungi regulate insect populations, curbing pests and protecting forests. In the Jurassic and Cretaceous, where herbivorous insects abound, this natural control maintains balance among animals, plants, and fungi. By targeting certain hosts, they reduce pressure on vegetation and, indirectly, preserve the diet of large herbivores. While parasitic fungi regulate insects, others, mycorrhizal, ally with roots. Under Araucarias, still present in the southern hemisphere, boletes and amanitas form ectomycorrhizae. This consists of a mantle of filaments wrapping around roots and insinuating between their cells, unlike endomycorrhizae, which penetrate inside. In this symbiosis, the fungus provides phosphorus, nitrogen, and micronutrients to the roots, in exchange for sugars produced by the plant. This collaboration increases trees' resistance to drought, deficiencies, and attacks. In the forests of the Jurassic and Cretaceous, it helps maintain stable ecosystems at the foot of the giant conifers. Boletes, with their thick stem and spongy cap, originate from more discrete ancestors but with the same role, weaving a dense subterranean network through their hyphae. Amanitas, including the famous fly agaric, also have their precursors in these forests. Less diversified, they already showcase traits that unite them in symbiosis with conifers. Boletes and amanitas thrive under conifers like Araucarias due to their ability to degrade resins and organic compounds released by these trees. In return, they receive the sugars produced by the needles. The visible mushroom is only the tip of a vast underground network connected to the roots and the result of long invisible work. In this context of biological effervescence, spore dispersion is crucial. These tiny reproductive structures, akin to seeds, only germinate in moist soil, rich in organic matter and connected to compatible roots. Once released, they travel via wind, water, or animals. During the Jurassic and Cretaceous, large herbivorous dinosaurs play a key role. By brushing against branches, trampling moss, or simply moving through vegetation, they carry spores attached to their scales, budding feathers, or rough skin. Even the partial digestion of infected plants can, through their excrement, seed new favorable sites. Beetles and other flying insects disperse spores by covering themselves while foraging or rummaging in litter. Attached to their carapace or moist areas, they travel to new sites, avoiding inbreeding and promoting fungal expansion. This relationship leads to coevolution. Insects and fungi mutually refine their adaptations. Ancestral cordyceps could transform an infected host into a gathering point, attracting other insects exposed to the spores. This mode of propagation, brutal but effective, regulated populations and promoted fungal diversification. The abundance of insects during the Jurassic and Cretaceous provided an ideal setting, maintained by the constant activity of the fauna. Flora, fauna, and fungi formed a tight ecological network in the Mesozoic. Dinosaurs, through their movements and feeding, influenced fungal populations. Trees relied on fungi to access rare nutrients, while fungi used animals and insects to disseminate their spores. These exchanges went far beyond predation or parasitism, constituting vital collaborations for biodiversity. Beneath the litter, mycelium intertwines with roots in soil aerated by primitive worms, terrestrial crustaceans, and other burrowers. Each cubic centimeter houses a micro-universe of fungal networks. During the Jurassic and Cretaceous, continental drift altered rainfall and oceanic balances, influencing soils, forests, and plant-fungi interactions. The rise of boletes and amanitas in Araucaria forests reflects a strong alliance. Fungi benefit from the conifers' resistance to harsh environments, while these gain in nutrition. The needle litter, acidic and rich in resins, is decomposed by specialized species that release minerals and promote renewal. The undergrowth thus becomes the stage for a true biological symphony. During the Jurassic and Cretaceous, pollinating beetles, ancestors of scarabs, disperse spores by consuming fragments or secretions, scratching the cuticle or digging tunnels. These behaviors fragment the fungi and multiply germination sites, integrating into an ecological network where each organism is linked to many others. In some small dinosaurs and primitive mammaloids, the occasional consumption of young mushrooms, rich in water or nutrients, could allow spore dispersion via excrement. In large dinosaurs, this dietary part remained small, but the mechanism is reminiscent of what is observed today in birds and large mammals. During the Cretaceous period, the first flowering plants appeared. The continental drift continued, and dinosaurs evolved, with some disappearing while others thrived. The first feathered dinosaurs marked the precursor to birds. On the ground, fungi and tree roots strengthened their alliance, colonizing more arid environments. Entomopathogenic fungi controlled insects, maintaining the plant balance. Coniferous forests remained sanctuaries of biodiversity. The dense animal diversity and vegetation provided fungi with an ideal environment. This created opportunities for symbiosis or parasitism, abundant organic matter, and conifers to form durable mycorrhizae. The mobility of insects, dinosaurs, and small mammals favored the dispersion of spores in these ancient forests. Fossils sometimes reveal fragments of fungi in amber, with filaments, spores, or parasitized insects, as well as alterations in arthropods linked to entomopathogens. Boletes and amanitas rarely fossilize, but roots bearing mycelial traces or an ectomycorrhizal sheath testify to the conifer fungi co-evolution. More than mere decomposers, fungi wove the vital links of Jurassic and Cretaceous forests, regulating balances, forging symbioses, and spreading biological diversity. Discrete yet essential, they left a lasting imprint in the soil and on the fauna. In these insect-laden forests, their actions, combined with those of dinosaurs and beetle pollinators, shaped ecosystems that would have been quite different without them. From 66 to 34 million years ago, a warm climate favored the rise of tropical forests, plants, insects, and microorganisms. Non-avian dinosaurs had vanished, making way for new species. Some fungi diversified to take advantage of these conditions, playing a key role alongside fauna in transforming vegetation. It is in this environment that thermophilic fungi and their adaptations to heat developed. After the extinction of the dinosaurs, the climate warmed until the Eocene thermal maximum. Humid forests expanded, offering fungi an abundance of plant matter. Thermophilic Escomycota, capable of degrading cellulose through their enzymes, exploited wood, leaves, and other debris to grow massively. Thermophilic fungi withstand temperatures above 35 degrees Celsius thanks to reinforced membranes and enzymes active at high heat. By degrading cellulose, they regulate carbon, accelerate decomposition, and release nutrients benefiting the entire tropical ecosystem, creating a virtuous cycle between heat, recycling, and biodiversity. Coprophilous fungi, which appeared with the rise of mammals, exploited the fiber-rich and mineral-rich excrement with specialized enzymes. They recycled this matter into soil fertilizer and spread their spores, resistant to digestion, via their hosts. This strategy allowed them to quickly colonize new areas in the Paleocene-Eocene. In the Eocene, ericoid mycorrhizae, resulting from alliances between fungi and heathers, enabled these plants to exploit acidic and poor soils. In recent volcanic zones or leach substrates, they colonized slopes and bushy forests. This underground cooperation shaped unique ecosystems and prepared the way for the establishment of other plant lineages. Nothofagus, or southern beaches, formed mycelial networks with their fungal partners, optimizing access to water and nutrients in exchange for sugars. This bond favored the expansion of mixed forests, whose spores spread by wind, water, or animals, enriching and stabilizing soils. In the Eocene, plant decomposition by fungi released compounds that circulated within ecosystems, while forests captured CO2. Despite a warm climate, this fungal bloom limited greenhouse gas accumulation, stabilized soils, and stored carbon. Alliances such as those between Nothofagus and ericoid plants illustrate this diversification. At the end of the Eocene, the climate began to cool, but fungi, including many Ascomycota and copro-dependent species, had already developed a strong heat tolerance through cellular defenses and a rapid metabolism. Stimulated by competition, plant growth, and the arrival of large mammals, they integrated into complex networks and, sometimes via insects, dispersed their spores. The soil moisture increased their biomass, a benefit that persisted into the early Oligocene. During the Paleocene and Eocene, the warmth accelerated the diversification and expansion of thermophilic fungi. Saprotrophs enhanced their enzymes to decompose wood, leaves, and dung, while symbiotic fungi multiplied their plant partnerships. Tropical forests teemed with mycelial networks, ensuring plant resilience. At higher altitudes, ericoid mycorrhizas colonized acidic soils, and false beaches thrived. Heirs of generations adapted. These fungi continuously transformed and recycled organic matter. From the Oligocene to the Miocene, the drier climate forced ecosystems to adapt. Grasses multiplied and colonized vast areas, offering new resources to fauna. Endophytic and mycorrhizal fungi reshaped the relationships between plants and soils, playing a central role in these expanding grassland environments. Mycorrhiza is a symbiosis where fungal hyphae extend the roots, improving water and nutrient absorption, while the fungus receives sugars. During the Oligocene and Miocene, this alliance allowed grasses to thrive despite the drought, paving the way for the expansion of large grasslands. In the grasslands, mycorrhizal fungi thrived alongside C4 photosynthesis plants, typical of warm and sunny areas. The glomeromycetes, adapted to these grasses, excelled at extracting minerals in poor soils, offering a decisive advantage. This alliance demonstrates the ability of organisms to adjust to changing climates. In the Oligocene and Miocene, endophytes, fungi living in leaves and stems without harming the host, evolved alongside the expanding Poaceae. Some, like the ancestor of ergot, produced toxic alkaloids, deterring insects and mammals. This chemical defense influenced the distribution of herbivores and the dynamics of grazing lands. The retreat of forests and the rise of savannas favored grasses, supported by mycorrhizal and endophytic fungi. Adapted to limited light and water, they fed large herbivores and modified the silicon cycle thanks to their phytoliths, which harden tissues, slow down mastication, and influence plant decomposition. By decomposing plants, fungi released silicon from phytoliths, making it available to soils and then, through leaching, to aquatic environments. In the arid savannas of the Oligocene and Miocene, this mineral circulation, stimulated by fungal action, supported fertility and influenced the diversity of silicon-dependent organisms. In the dry savannas with C4 grasses, a specialized megafauna coexisted with saprotrophic fungi, exploiting the grassy litter. The short but intense rains stimulated their growth, accelerating nutrient recycling. Microbial competition reinforced fungal defenses, complicating underground interactions, and making the savanna an ecosystem finely orchestrated by invisible actors. In the burgeoning savannas, alliances between Poaceae and endophytic fungi allowed colonization of more arid environments. By enhancing drought tolerance, protection against pathogens, and underground resource storage, They favored the rise of African, Asian, and South American savannas. Over time, these symbioses influenced plant distribution and ecosystem structure. At this time, fungi developed spores capable of surviving years in dry soil, germinating with the return of rain. They then resumed decomposition, nutrient exchanges, and defense production. Several species tolerated temperatures exceeding 35 degrees, illustrating their exceptional resilience to the extreme conditions of savannas. In retrospect, the Oligocene-Miocene marked a turning point in the relationship between fungi and grasses. In a dry climate, these plants adopted C4 photosynthesis and accumulated phytoliths, while their fungal symbionts ensured their survival. This alliance stimulated the rise of savannas and the diversification of fauna. Endophytes, mycorrhizae, and saprotrophic fungi shaped the soils, protected the plants, and regulated nutrient cycles. Thus, this period established the foundations of resilient landscapes, molded by the cooperation and plasticity of all the organisms involved. The early Pleistocene, from 2.6 million to 300,000 years before our era, alternated glaciations and milder periods. Glaciers advanced and then retreated, opening corridors where plants and animals took refuge. These variations transformed the distribution of ecosystems. From grassy steppes to sparse forests, megafauna occupied contrasting habitats. In this changing environment, fungi played a key role. Some, like polypores, decomposed dead wood. Others, pyrophilic, recolonized soils after fire. Their hyphae transformed organic matter and released essential nutrients. Even in extreme cold, their robust enzymes ensured continuous activity, and during thaws, the humidity favored their multiplication and the dispersion of their spores by the wind. The megafauna, including mammoths, shaped environments through their trampling and grazing. Their excrement, rich in fiber, nourished microorganisms, including fungi. Polypores exploited trunks felled by winds or weakened by frost, breaking down wood and releasing nitrogen and minerals. Despite the cold, some fungi adapted their metabolism to survive partial freezing, ensuring nutrient circulation even in winter. During thaws, their activity accelerated, rapidly transforming dead plant material and producing large, leathery, fruiting bodies. The sparse forests of resilient conifers offered a stable habitat for these fungal communities, supporting a precious biodiversity. In the early Pleistocene, fire, triggered by lightning or amplified by drought, was a recurrent phenomenon. During warmer periods, the expansion of vegetation cover increased combustible biomass. In the dry season, withered grasses and shrubs facilitated the spread of flames. Although seemingly destructive, the fires favored new colonizations. Pyrophilic fungi sprouted in the ashes, taking advantage of the nutrients released. The charred wood, ignored by herbivores, became a refuge for these species, whose filaments penetrated the burned layers, breaking down the material and returning essential chemical elements. This recycling stimulated the regrowth of pioneer plants, attracting megafauna and dispersing spores far and wide, making fire a driver of biological diversity. Glacial droughts appeared when atmospheric humidity dropped, with water trapped in vast ice sheets. The cold, dry winds thinned the vegetation and reduced forest cover, leaving dried stems that favored fires. Pyrophilic fungi then took advantage of the burned surfaces, their heat-resistant spores ensuring recolonization. The decomposition of charred wood and the mineralization of ashes hastened the return of pioneer plants. Less adapted to fire, polypores remained on the woody fragments spared. Large herbivores crossing these zones dispersed spores attached to the charred barks. This cycle of drought, fire, and fungal recolonization shaped mosaic ecosystems, mixing open spaces and preserved groves, promoting the diversification of fungi and plants. Sporopollen indices, obtained from analyzing the proportions of spores and pollen in sediments, offer precise insights into this period. They reveal the frequency of fires, the evolution of vegetation, and the abundance of decomposer fungi. A decrease in pollen often indicates a retreat of vegetation caused by cold or drought, while a rise in spores signals intensified decomposition. Pyrophilic species distinctly mark charred layers, and examining partially burned organic matter allows assessment of fire intensity and regeneration speed. These observations distinguish glacial phases with low vegetation cover from milder periods where flora and fungi flourish. They illustrate the close links between climate, vegetation, fire, and fungi. Dansgaard-Oeschger cycles are rapid climate oscillations occurring in the heart of glacial periods, highlighted by Greenland ice cores. During these temporary warmings, vegetation briefly thrives, stimulating biomass production and providing more resources for decomposer fungi. But the sudden return to cold puts these communities under strain. In continental sediments, spikes in fungal spores are observed, followed by a significant decline. Fires also follow this rhythm. Wet phases limit fires, while droughts increase the flammability of the plant cover. This cycle makes fungi true sentinels, adapting their distribution and activity, thus influencing soil balance and flora regeneration. In this changing landscape, megafauna serve as vectors for many fungi. Large herbivores traverse vast territories, transporting spores on their fur and churning the soil by fragmenting plant litter, creating new niches. After a fire, a herd trampling the charred ground can bury pyrophilous spores in the top layers. Their nutrient-rich droppings also promote the proliferation of specialized fungi, essential for recycling organic matter and releasing mineral elements for the flora. This coevolution, active even in the frozen margins of the early Pleistocene, enabled life to persist in extreme conditions. Polypores stand out for their ability to decompose lignin, which stiffens wood. In the early Pleistocene, trees weakened by cold or toppled by winds formed a vast reservoir for them. The decomposition of trunks released nutrients, enriching the soil and promoting other life forms. Pyrophilous fungi, on the other hand, colonized the charred layers of burnt stumps or grasses. Together, these decomposers profoundly reshaped ecosystems, attracting xylophagous insects and predators. True discrete architects, they opened successive niches, while the alternation of cold, drought, and fire multiplied opportunities for renewal, making fungal communities ever more adaptive and contributing to the complexity of ancient environments. Despite a harsh climate, life concentrates around strategic points: open water areas, sheltered valleys, and migratory corridors. Fungi exploit every opportunity: a fallen tree near a watercourse, a localized fire, or the passage of animals. In conifer forests, the resins require specific enzymes, which some polypores possess, accelerating wood decomposition. During warmings, the flora diversifies and multiplies hosts. When the cold returns, only resistant fungi persist. Sporopollinic data then reveal rapid changes, proof of great plasticity. Fungal meiosis constantly produces new genetic combinations, accelerating the evolution of already resilient populations. Thus, the landscape mosaic contains multiple adaptation scenarios, dictated by the rhythm of the glacial cycles. At the end of this long period, up to about 300,000 years, consolidated fungal networks stabilize ecosystems.